Hello, everyone. Good afternoon or good evening or good morning, wherever you are. And welcome all of you to today's special webinar. My name is Shin. I'm an assistant professor in the Department of Surgery at Baylor College of Medicine in Houston, Texas. I am very excited to have all of you here today. And I know we'll have busy schedules, especially at the end of the year, holiday season. So I greatly appreciate all of you being here today. And I'm very excited because today we're going to explore the fascinating field of cutting-edge imaging system, intravital imaging. And intravital imaging technology has been a game changer for many researchers, enabling high-resolution imaging and real-time monitoring of cellular behavior in live animals. And IVM technology based in South Korea has been instrumental in supporting this work with demonstrations at prestigious institutions like Harvard Medical School, Brigham Women's Hospital, UMass, and Johns Hopkins. And I'm really hoping to add Baylor College of Medicine to that list or any other institutions in the Texas Medical Center. And my research focuses on developing drug delivery systems and surgical implants. So I'm very much interested in learning about this system because we think it'll be very useful for our research program. So now I'd like to introduce you Dr. Pilhan Kim. He's an expert guide for today's session. He is a professor at the Korea Advanced Institute of Science and Technology, or KAIST. So he's been instrumental in the field of intravital microscopy and imaging techniques. And I mean, with his impressive academic background, he was the research fellow at Harvard Medical School from 2005 to 2010. And in 2017, he ventured outside of the academia. I mean, in addition to his academic appointment, he founded this IBM Technology. It's a pioneering company dedicated to advancing intravital imaging systems. So please help me welcome Dr. Kim. The floor is yours.
Hi, thank you. Thank you very much for the kind introductions. And then I'm very flattered. Okay, let me share my screen. Okay. All right, so I hope everybody can see my screen now. All right, so let me start. Okay, thank you very much for joining us today. So today I will talk about the real-time intravital microscopy system for various preclinical research. This intravital microscopy is based on laser scanning confocal and two-photon microscopy, and it's capable of in vivo cellular level imaging of various internal organs in a live animal. Also, it can be a very useful tool for the in vivo imaging analysis of drug delivery, drug efficacy, and drug distribution in a live animal. Okay, so I have a COI, of course, and then this is my career. So I'll just skip it. Okay, so yeah, this is the introduction slide, what you can expect to obtain by using intravital microscopy compared to other imaging modality, such as X-ray, PET, CT, MRI, or whole body optical imaging systems such as IBIS. Compared to that, intravital microscopy is capable of microscopic scale cellular level video acquisition in a live animal model in vivo. So this particular example is obtained from the lung of a live animal in vivo. And then in this example, the green color shows the blood flow visualized by FIT spectrum intravenously injected. And also red color shows the neutrophil inside the vessel. And then this neutrophil is fluorescently labeled by antibody conjugate, which we call Li6G, Alexa-647. So in this example, so Li6G is the surface marker of the neutrophil. So what we prepared the antibody of anti-Li6G, and then we conjugate them with the fluorophore, the far-red fluorophore, Alexa-647, and we intravenously injected those antibody fluorophore conjugate at 30 minutes before the imaging, and it nicely labeled the neutrophil inside the vessel. And after that, by using intravital microscopy, we obtain this video in the lung of a live animal model. And then this video I will explain a little bit more later. And then let me stop it here. And then also, basically, intravital microscopy is based on laser scanning, fluorescence, confocal, and two-photon microscopy. So it can record, it can acquire, it can detect the fluorescence signal in the live tissue. So it means if we can label the drugs fluorescently, then we can image the drug distribution in a live animal. So this is one example. So this video I will start now. And then this is the real-time video obtained from the liver of a live animal model. And in this this live mouse model is type 2 GFP mouse. And then in this mouse, every endocellular cell strongly express GFP under the promoter of type 2. So this green color is the endocellular cell in the liver, in the sinusoid capillaries. And then when I start this video, you can see the flow of the fluorescently labeled nanoparticle intravenously injected through the tail vein. So let me show it again. So I hope you can see this video very well. So this red color is the flow of the fluorescently labeled nanoparticle intravenously injected. And then this is how it looks like in the liver in real time. So if you look at carefully, you can see this flowing nanoparticle inside the liver capillaries. And then also at the same time, you can see how this nanoparticle is accumulated inside the liver capillaries, liver sinusoid capillaries in real time. Okay, so in short, this intravital microscopy can acquire this live cellular level video in a live animal model with some molecular information by using targeting moiety such as antibody. Okay, so in my lab at KAIST and also in IBM technology, we have been able to image almost every organ and tissue in live animal model, mostly mouse model. And all these images were obtained from the live and staged mouse model. So you may be able to find the organ of interest for your research, I believe. So we have been able to image the bone marrow by brain tumors. And then again, all these images were obtained in vivo from live animal model. And then we also have imaged the retina, skin. This is the hair follicle in the skin. And then we have imaged the various tumor model. This particular one is a colorectal tumor model implanted in the cecum. And pancreas, spleen, stomach, small intestine, colon, and then various adipose tissue, prostate. And then this is the image obtained from the placenta of a pregnant mouse model, lymph node, memory tissue. And this is some image obtained from the kidney after cell transplantations. This is kidney, muscle, liver, heart, lung, thyroid, and so on. So this list is keep increasing. So at this moment, I cannot put every images in here now. But anyway, so this is the slide. We can use intravital microscope to image almost every organ and tissue in a live animal model in very high resolution. So this is some selected publication list for each individual organs. So today I will mostly talk about this dual mode intravital confocal and two-photon microscope. This one, the CMS3. And this is a very much optimized system for in vivo several level intravital imaging of a live animal model. And then I show you those two examples previously. And it's quite small. It's only 630 millimeter in width and 890 millimeter in length. And the height is 490 millimeter. So it's quite small. So you can put this microscope easily inside the lab or inside the animal facility. And then also this is capable, well, let me explain a little bit more later. And then it's also capable of two-photon imaging with 4 detectors. So it's capable of 4-color simultaneous two-photon imaging. Also, it's capable of 4-color simultaneous confocal imaging as well. And then inside this microscope, we use very high-speed laser scanner for real-time imaging with the speed of 30 to 100 frames per second with 512 by 512 pixels. And then this high-speed imaging capability is very important to implement automatic live tissue motion compensation function, which is very critical for live animal imaging. Okay, so let me show you just one quick example. So this is the video of setting up for the mouse for the stabilized lung imaging. So if you look at here, you can see this anesthetized mouse model. This mouse is intubated in here. And then this is the lung. So we opened the chest and then exposed the lung. And then in this chamber, if you look at here, we have a small suction hole here. So we can suck out the air through this suction tube and build up the negative air pressure, very gentle negative air pressure. And then by doing that, we can stabilize the lung surface against this transparent cover glass on top of it. So if you look at it, yeah, like that. So this lung surface is stabilized. When in macro scale, but even after this macro scale, stabilization using suction, still we have some residual, tissue motion in microscopic scale. So this is a real-time video obtained from the lung of live anesthetized mouse model after this suction-assisted lung stabilization. So the field of view is 250 micrometer and 250 micrometer here. So you can nicely see the capillary in the lung. And then this red color is intravenously injected fluorescent labeled red blood cell. So we can see this RBC flow in the lung capillaries. But you can also notice that we have some, tissue motion, in the lung tissue, even after this mechanical stabilization using suctions. So to obtain high resolution images, stabilized high SNR, high resolution imaging, we need to deal with these tissue motions. And then that's why we need this very high speed imaging capability. So this is another example. This video rate images obtained from the kidney of a live mouse model. And then this is a very typical tissue motion you will see when you try to do in vivo intravital imaging of a live mouse model. So as you can see, so we have a very huge, very significant tissue motion. So the way to mitigate this motion, tissue motion, is frame by frame motion compensation. So what we do, which is very straightforward, we just record the video in very high speed, faster than 30 frames per second, and then we analyze each frame, like frame 1, frame 2, frame 3, and frame 4, and then we can see how this cell is moves around due to tissue motion, and then we do the frame by frame image registrations to correct this tissue motion from this video. And then we just average this video and then to generate this high resolution, high SNR images from the moving tissues. Okay. So you can easily compare the result with motion compensation, frame by frame registration, or without any motion compensation functions. So if your microscope is slow, like 1 frame per second or 2 frames per second, then you will end up with very blurred images. Okay, and then so definitely you can see the advantage of real-time imaging and then frame by frame motion compensation to acquire these very high resolution images from the moving tissue in the live animal model. So in our microscope, we implement this motion compensation function to be automatic. So what you need to do is just turn on this motion compensation function and press the image capture buttons. And in just a couple of seconds, you can generate this motion compensated frame by frame image registered images from the moving tissues. Okay, so that is the key capability to allow us to obtain these very high resolution images from various tissue and organ in a live animal model. Okay, so let me come back to lung imaging again. So I already explained about these setups. And then this is a 10-minute video obtained from the lung of a sepsis mouse model. Well, this is a published work already. So in this 10-minute video, what you can see is this neutrophil, this red color neutrophil is very active. They are, they are very actively moves around inside the vessel in the lung after with in sepsis conditions. And then if you look at in here, you can see roughly 7 neutrophil, and then they start to contact each other, and then suddenly it make a huge cluster. Well, not huge, just a 7-cell neutrophil cluster, and then those neutrophil cluster, it can physically block the arteriole here, and then disturb the microcirculation in the lung. So this 10-minute video, clearly shows that the neutrophil is the major player that blocks the vessel in the lung of a sepsis mouse model. So like this, okay. And then let me just quickly show you how we implement this lung imaging in the live animal model. So we first need to intubate, and then we connect the mouse to the mechanical ventilator, so to keep the mouse to breathe air while we do this surgery. And then we open up the chest, expose the lung, and then stabilize with the suctions like this. And then we put the mouse into our microscope, and then put it under the high magnification objective lens, and then do the laser scanning, and then obtain the live images, which I showed you before. And then we use motion compensation function to obtain these high resolution images from the moving tissue, moving lung tissue. Okay, and then we can use the same principle for the heart imaging as well. So as you can see in here, we use suction again, and then we can stabilize the surface of the beating heart in the live mouse model. And then only this window, it has just 2 millimeter open area. So only 2 millimeter surface of the heart is stabilized with the glass in here. And after that stabilization, we can also, again, we can observe this very high speed phenomena such as flowing cell in the heart surface. And then also last year, we developed some additional tool AI processing framework for the noise reductions, for denoising of the real-time videos obtained from the live animal model. So this is published work again. And then on here, on the right-hand side, this is the raw video. This is 30 frames per second real-time video obtained from the skin of a live mouse model. And this particular example, we use H2B-GFP Roger 26 MTMG mouse model. So basically in this mouse model, every cell nuclei express GFP, and then every cell's membrane express TD tomato. So we can easily see the cell nuclei and the cell membrane. And then this AI model is very useful to remove the noise from this real-time video. So on the left-hand side, this is the result. So this is not tissue motion compensation. So this is a post-processing algorithm, and then it can be applied to any video in unsupervised manners. And then this is the result. So we can easily increase the SNR more than 2 orders of magnitude. So another example. So this is the real-time video obtained from the kidney. And the left-hand side, this is AI processed denoised real-time videos. And then another example, this is from the muscle. And then you can nicely see this flowing red blood cell in this large vessel in the muscle. Okay, so from now on, let me quickly show you, several more examples how we use intravital microscopy for in vivo imaging of cells and drugs in a live animal model in various tissue and organs. And this is our preparation for the lymph node imagings. And so again, the mouse is anesthetized. And then we catheterize the tail vein using the catheter. And so by doing that, we can inject the cell through the tail vein. And then in this example, we made the incision of the skin and exposed the perforated lymph node, and then cover the perforated lymph node with the transparent cover glass. And then this is the 2-hour time-lapse video obtained from the lymph node. And in this particular example, what you are looking at is a T cell in red color and B cell in green color. And blue color is another cell called FRC. It's a fibroblastic reticular cell inside the lymph node. And then what you're looking at is extravasation of intravenously injected T cell and B cell in the high-natural venue in the middle of the lymph node. And then this arrow, point out the extravasation site of these two kinds of lymphocyte. So if you look at here, so you can see this B cell comes in, and then it interacts with the FRC, and then it comes out, right? And then later the T cell follow the exactly same path and then comes out from the vessel. So this 2-hour video nicely shows the tracking, the extravasation tracking of T cell and B cell inside the lymph node. So this video nicely shows that the intravital microscopy can be a very useful tool to analyze the cell movement and their tracking, and then for the tracking of their movement inside the live tissue. Okay, and then let me show this example again. So I already showed you. This is the real-time intravital imaging of intravenously injected nanoparticle through the tail vein, and then to analyze their accumulation in the liver sinusoid in real time. So this is an AI processed video actually. And then again, we use type 2 GFP. And then in this mouse, it expresses GFP in vascular endothelial cell. So we can easily see the liver sinusoid in green color. And this is the preparation. So we anesthetize the mouse, and then we insert the lactite probe to monitor their body temperature. And then we use a heating pad to keep the mouse body temperature to be stable during our imaging. And then again, we use tail vein catheter to inject the nanoparticle through the tail vein. And then when I start this video, that's when we do the intravenous injection of this nanoparticle. And then I hope you can see. So if you look at on the right-hand side, you can see this nanoparticle, flows into the liver sinusoid, and then they quickly attach to the liver sinusoid, and then accumulating in certain area in here, for example. Okay, so this is how it looks like when this is what happens when you do the nanoparticle injection, and then how they accumulate and distribute in the liver in real time. Okay, so this is just 1 minute real-time video, but we can extend our imaging time by using other technique such as imaging window. And then this is another published work several years ago. And then in this particular example, we use dorsal skin pole chamber. This is a very well established technique that can implant this metal imaging window on the back of the nude mouse model. And then when we implant this dorsal skin pole chamber on the back of this nude mouse, we inject the GFP expressing human cancer cell line, MDAMB231. This is a triple negative human breast cancer cell line. And then it expresses GFP. And then we injected this cell line in the middle of this dorsal skin pole chamber. And then we let this mouse to grow the breast cancer for 2 weeks, more than 2 weeks. And then we bring the mouse, anesthetize it, and then we inject the nanoparticle through the tail vein. And then we observe their accumulation inside this chamber at 2 hours and 6 hours and 24 hours after the injections. And then because we are using this dorsal skin pole chamber, we can find the exact same location inside this mouse model at different time point. And then to find out the same location, we label the vessel by using antibody conjugate. So again, the CD31 is the surface marker of the endothelial cell. So we prepare anti-CD31 antibody, conjugate with the fluorophore, and then we do IV injection of that antibody conjugate to label the blood vessel. So let me remove the green colors. So if you look at the blue color, so we can easily identify this is the same vessel and same microscopic areas. And then when you look at these red colors, you can see how this intravenously injected nanoparticle, distributed inside of these tumors. So 2 hours, and we don't see many. At 6 hours, you can see, a lot more nanoparticle, mostly inside the vessel, and then some in here and some in here. And then finally, at 24 hours, you can see the delivery of this nanoparticle to the real tumor cell. For example, here, and also here. So by using this imaging window and using intravital microscopy, we can find out the exact same location in a live mouse model over several hours or several days, and then analyze the delivery of the nanoparticle or other type of drugs to the target cell. And then in this case, the target cell is the breast cancer cells. Okay, and then I like to say that we have imaged the nanoparticle, but we can also image other modality of the drugs, such as antibody drugs, exosome drugs, and the peptide drugs or RNA drugs, and even the cell itself as well. So that was just 24 hours imaging. And then here, I like to also say that the beauty of intravital microscopy is the mouse is alive during the imaging, and then we can save the mouse after each imaging session. So it means we can repeat the intravital imaging in the same mouse model at different time point. And this is another example of that. So this is the longitudinal repetitive wide area intravital imaging of cranial bone marrow in the mouse model after bone marrow cell transplantation. This is day 1, day 3, and day 4. And this red color is the transplanted cell. And this is a very wide area mosaic images. And then we also use the vessel to look at the same site. So if you look at here, day 1, not many. Day 3, we can see the accumulation of the cell in the central sinus. And day 4, we can see the proliferation of this transplanted bone marrow cell like this. And then this is how it looks like at day 3. So we can see their migration and proliferations. And then again, we can repeat this imaging many times. So this is from day 2 to day 10. And then we can find the exact same location again. So at every day. So day 3, you can see some cells here. Day 4, more, 5, 6, 7. So this cell, proliferate very rapidly until day 7. And suddenly at day 8, they disappears, right? Look at here, they disappear. And then surrounding area now have, more cells. Okay. And then by looking at the vessel, we can find out the, exact same location, as you can see in this example. And then we can repeat this intravital imaging to see how the transplanted cell behaves in the live animal model over a long period of time. And then to find the exact same location, we normally label the vessel like this in this example. And then we start from the lower magnifications. So like a Google map. So we first look at the low magnification and then identify the large vessel. And then we just gradually zoom in to find the exact same location in microscopic scale in this example, as shown in this example. Okay. So this is another example. So this is the ongoing work. So in this work, we implant this ring-shaped imaging window on the side of the mouse model to image the pancreas and spleen. Okay. And then at day 0, when we implant this imaging window, we inject this mouse pancreatic cancer cell, PANCO2 cell, into the pancreas. So basically, we are making the orthotopic pancreas tumor model. Okay. And then we can just repeat our imaging, as you can see in here, and then see how this pancreatic tumor cells, grows. As you can see in here, day 9 and day 14, you can see the pancreatic cell grows a lot. And then you can also see this blood vessel labeled by CD31 antibody. And you can see how this tumor vessel changes over time. And again, by using this imaging window, we can find the exact same location from day 9 to day 14. So I like to emphasize that again, this is the exact same location in the same mouse model. So every day we analyze the mouse, and they find the one locations, and then recover the mouse, put it into the cage, and the next day we bring the mouse again, anesthetize it, and then find the same locations. So, in this example, so you can see how this pancreatic GFP expressing pancreatic cells grows from day 9 to day 14. And then you can also see, if you look at carefully, you can also see very dynamic, blood vessel changes together with the tumor cell growth. So if you compare day 10 to day 11, so you can see, if you look at here, you can see the growth of the pancreatic cells. And then also you can see this enlargement of this particular vessel in just one day from day 10 to day 11, right? And then this part also changes over time like this, okay? So this is the very one good example to show the interaction between the cancer cell and then tumor vessel while they grows in vivo conditions. Okay, so this is my final example. So this is, I like to introduce this cranial imaging window surgery. This is again, very well established technique for longitudinal repeated brain imagings. So basically we remove the small part of the skull like this, and then we put the transparent glass on top of it. And then we sealed up the exposed skull area with the dental cement like this. So this is the images. This is the photo after this surgery. And then after this cranial imaging window surgery, we can keep the mouse to be alive for more than several months, even to 1 year. So basically, we can keep the mouse alive, and then we can repeat the brain imaging in this mouse model. And then this is the one example. This is wide area mosaic G-stacks images, starting from the meninges to the brain cortex. So if you look at here, this is the meninges, and then you can see this vessel in the brain surface, and then you can see how this vessel is penetrating into the brain. So let me quickly explain one of our work. So because we can identify this penetrating arteriole, we can target those arteriole and then make a photothrombus in those arterioles. So basically we are making the mini stroke in the mouse model, which we call microinfarctions, by using a photocoagulation agent, lojbenga. And after that, we just follow up what happens from day 0 to day 30. And this yellow square, shows the infarction, this mini stroke site. And you can see the expansion of the tissue, brain tissue, which suggests the edema. And then from day 2 and day 3, it picked it. And then day 5, it goes back to the original size. And then after that, we can see the tissue shrinkage, suggesting the cortical tissue atrophies due to these infarctions. And then when you look at the vascular leakage at day 3, you can see this red color is TRI tissue dextran. And then you can see it leaked out. Okay? When you compare to day 0 and day 3, you can easily see the leakage of this circular Dalton dextran into the brain. So suggesting the BBB dysfunctions in terms of vascular leakage. And at day 5, interestingly, this vascular leakage was reversed. So again, this BBB dysfunction is resolved. So it leaked out at day 3 and then recovered at day 5. So that's why we see the tissue expansion at day 3 and its recovery at day 5, as you can see in here. And then we want to see what happens during that point of time. So from day 3.5 to day 4.5, this is 24-hour continuous imaging of the brain vessel. And then we identify this pericyte, this red color pericyte, extend their process along the endocellular cell, along the vessel. So if you look at here, it enacts their process like this. Okay, and then this is how this vessel recover their vascular leakage dysfunctions. Okay, so I think this is one good example how we can use intravital microscope to connect the phenotype of vascular leakage and then how the cells behave during this change of their dysfunction, physiological dysfunctions. So intravital microscopy is capable of 3D cellular level imaging in a live animal model in vivo. And then it can also provide molecular and functional information as well. So as I showed in those few examples, this intravital microscopy can enable a dynamic 3D imaging of various cellular level dynamics in different organ in a live animal model. And then we can observe the cell trafficking, cell to cell or cell to microenvironment interactions inside the live animal model in vivo. And also for the drug development, this intravital microscopy can enable a direct in vivo drug imaging analysis. So we can analyze the drug delivery to the target cell, and then also we can analyze the drug efficacy in the target cell after the delivery, and then validate the mode of action of the drugs we are developing in a live animal model in vivo. So we can do this kind of observation in every organ and tissue in a live animal model. So for following 10 minutes, I like to show several key features of our microscope systems. Okay, so again, this is a single box, full packaged microscope, and capable of 4-color 2-photon imaging and 4-color confocal imaging by using just the same microscope. So we can easily switch from confocal imaging mode to 2-photon imaging mode by using same microscope and then observe the same site. And for 2-photon imaging, we have 2 options of laser system. So one is using compact wavelength fixed femtosecond pulse laser module inside the microscope, or we can connect the conventional tunable femtosecond pulse laser system through the optical port behind its microscope box. Okay, so let me just quickly show what you can expect to obtain from the live tissue by using 2-photon imaging mode. So this is the G-stack images obtained from the skin of a live mouse model by using our wavelength fixed femtosecond laser module inside the microscope. And then this is a 4-color simultaneous detections. So you can see 4-color green color is GFP expressed in the nuclei. Red color is the TD tomato expressed in the cell membrane. And then blue color is another dye intravenously injected through the tail vein. So basically we are looking at the blue color is the blood flow. And the white color is collagen in the tissue, which visualized by the second harmonic generation signal generated by the 2-photon femtosecond lasers. So if you are familiar with the skin microanatomy, you can see the cell nuclei between the dermis and epidermis and dermis. And then now this is the dermis, the G-stack images. So you can white color dermal collagen, and then you can see the hair follicle. And then going deeper, you can start to see this micro capillary and the capillary endocellular cell nuclei. And then if you go deeper, then you can see, and then here, you can see this is a lymphatic vessel and then large vessels. So this is the skin. And this is another example of the muscle. So in this particular example, so you can nicely see the fascia in the beginning, and you can see the myocyte and the multinucleated myocyte, and this sarcomere myofilament visualized by the second imaging signal, as you can see here. And then you can see the capillaries and the large vessel inside the muscle. That was a single field of view. And then what you can do is our microscope is capable of automatic wide area mosaic images. So like this one. So we take a wide area image, like a tile scan images, and then we can automatically stitch those tile scan images and generate this wide area images, as you can see here. And this is pancreas and wide area images of the pancreas. This is kidney. You can see this tubular structure of the kidney, and this wide area images. And then, as I explained to you before, our microscope is capable of very high-speed imaging. So for now, it's capable of 50 frames per second to 100 frames per second as options. And then this high-speed imaging capability is very important to implement automatic live tissue motion compensation function, as I explained to you before. And then by using our high-speed imaging capability, we can directly see the flowing cells in the live animal model, as you can see in this video, this. And then we can do this frame by frame motion compensation to compensate the motions of the tissue and generate this high resolution, high SNR images from the live animal. And then also we are providing the heating system for the mouse model. So this is how we do it. So we use lactite probe inserted through the anus, and then we feedback control our heating plate. So to keep the body temperature to be stable during the imagings. And then we also have a glass heating system to heat up the glass covering the exposed tissue. And then we have additional temperature sensor, temperature indicator sensor to monitor the local tissue temperature during the imagings. And then we have this tablet for this temperature control. Okay. And then we have this sliding door. So when you open up these sliding doors, we have this motorized motorized stages, 3D stages. So, and then it has a sliding mechanics inside, so we can slide out our stage and then put the animal model on the plate, on the heated plate, and then put it back, close the door, and do the imaging. So like this. And then we can also use gas sanitization system, such as isoflurane vaporizers. And then we have a 2-tube adapter on side of our microscope. So this is gas in, gas out, and then we use this head mask to keep the mouse to be anesthetized during our imagings. Okay. And then we have the full solution of the imaging window kit for the various tissue and organ. So for like the cancer models, heart, lung, brain, other abdominal organ like liver, kidney, spleen, pancreas, and so on, and the memory tissues. So we already made this all small device and apparatus optimized for the long-term imaging of internal organ in a live animal model. So that's very important for all this organ imaging in a live mouse model. Okay, and then our microscope is capable of live animal in vivo imaging. Well, in addition to that, it can also be used as a standard microscope for slide imagings. So this is one example. So we have an adapter for the slide imaging as well, and then we also have our own image processing and visualization software, which we call iBeam Studio. And this iBeam Studio software is capable of handling 2-dimensional, 3-dimensional, and 4-dimensional imaging data obtained from the in vivo tissue, in vivo animal model, or just slide scans as well. And this is one example. This is the whole brain scan very high resolution image scan images like this. So this is tile scanned images, and then this is the automatically generated switched wide area images. And then also, this software can handle the 3D imaging data. So for example, this is the optically cleared brain sections. And then this is the G-stack images with 2 millimeter depths. And then this is the rendered data set, 3D rendered videos generated by using our studio software. And then it can also deal with the 4-dimensional data. So basically, we obtain the 3D G-stack images at different time point, and then we can observe how the cells moves around in 3D in vivo tissue, as you can see in this example. Yeah, and then for your interest, we are also developing another system. So this is a miniaturized confocal 2-photon microscope, which attached to this articulating robotic arm. Okay. And then also, it's capable of very high resolution, subcellular resolution images of the live tissue. And then, this is still ongoing development, but we can easily, move our microscope attached to the robotic arm. So you can put it in any direction, in anywhere in the large animal. So the main purpose of this system is to image the large animal, such as the pig, dog, or non-human primate as well, and even for the human as well. And then we recently, this is another ongoing work. So we have our own AI framework to transform the fluorescence imaging into the histological images, as you can see in this example. And then it can be a very useful tool for the tumor margin detection during the surgery. Okay. All right. So to finish, if you are interested in our technology, please visit our webpage, www.ivimtech.com. You can find out about our microscope. And then also we have many information about what kind of a digital model you can image it by using Intravital microscope, and what kind of quantification you can do by using Intravital imaging data. And also, we uploaded many webinar series on our webpage with specific topics. So for example, like peptide drug imaging, or exosome drug imaging, or GI tract imagings, or some so if you have some specific keyword for your research, you may be able to find the related webinar for that research topics. And then also in South Korea, we have our own imaging animal facility, SPF animal facility, in-house animal facility, and in-house imaging labs. So we also provide the contract-based research service as well. So we can do just one-stop solutions. We can provide one-stop solutions. So this is just 2 examples of that, our research service. So our customer send us their drone material, and then we tested that material for them by using a live animal model. So on left-hand side, this is the exosome drug delivery monitoring example. So this red color is the exosome, and it's 10 minutes and then 30 minutes after IV exosome injection through the tail vein. And then we can see how this exosome was delivered to the target cell, like this, at 10 minutes and 30 minutes, this exosome was internalized into the cell. And on the right-hand side, this is a 2-day observation of the antibody drugs, again, intravenously injected. So upper panel, this is control drugs, so they don't come out from the vessel, and then they just disappear at 2 days. But this lower panel shows the delivery of these antibody drugs to the tissues. Okay. All right, so IBM technology, we are providing this confocal 2-photon microscopy-based Intravital microscopy system with advanced functions such as motion compensation, and then animal model maintenance and control in terms of temperature and energizations. And then we also provide the imaging service. And then we have our own specialist team with more than 10 years of experience in intravital imaging. So we can provide very in-depth consulting and technical support to optimize and design your intravital imaging experiment. And then we also provide the training service for the intravital imaging, such as surgical techniques and other labeling, and then handling of the live animal model for intravital imaging. So we provide those, basic and advanced training service as well, and also standard technical service to maintain the microscope system in your lab. All right, so thank you very much for your attention. Then if you have a question, I can answer it.
Thank you so much, Dr. Kim. That was so awesome. I'm not gonna ask you how big your your slides are with all the videos, but that was awesome. We do have a question in the chat, but I think you already answered. But Edwin
Okay.
asked, does brain imaging require a craniotomy?
Yes, it does. Yeah, it's shown during the talk. Yes.
So if you have a question, feel free to ask. I'm not sure if you're able to unmute yourself and ask questions yourself, but feel free to utilize the Q&A box. But I have a question about the kit that you showed for various organs. Are those reusable? You can
Yeah,
or those
it can.
autoclavable?
Yeah, it's reusable. So yeah, we do the autoclave and reuse it.
I see. And you showed all these cool images, and but you had a retina image, and I've tried regular intravital system to image retina, but it was very difficult
Yeah.
to to even focus. And of course, the breathing, the software can compensate the movement, but
Yeah, that's one issue. So yeah, here I need to clarify that to image the retina, we need a modification of our standard microscope system.
I
So
see.
so for the retina, we need a modification, additional modification to make the focus on the retina through the
But
eye.
have you imaged the corneas?
Yeah, we did actually. Cornea is easy,
Easy, because
Yeah.
it's right there.
But to image the retina, we need to look through the lens
Lens, yes.
in the mouse eyes. So we need additional lens to compensate, additional optical lens inside our microscope to compensate those mouse lens.
Yeah, the quality of your images are amazing. They're very clean, very sharp. And I know the advantage of using the Intravital system is reduced in background noise.
Yeah.
But I personally experienced, it could be my technique, but I've experienced still like I've seen the background
What kind
with
of
our
background?
two photon images. But yours is, I mean, I'm amazed how sharp and clean your backgrounds are. I mean, you virtually have no background.
Well, yeah, well, yes, well, I don't know. So it's all related with your signal level, basically. So if you can, so yeah, the key is, so one thing is we are using very sensitive detector in our microscope. And another thing is you need to presently label your target cell brightly, then you can reduce the background.
Yeah, so for the organs or targeted tissues you've experienced, do you also do you like provide like preparation support, like
Yeah.
tissue prep or
Yes, we
staining
do.
support?
Yes, we do. So we have a labeling kit for the antibody or cells
Oh,
for intravital
fantastic.
imaging. Yeah. And then I have been worked on intravital imaging almost 20 years. So I have tried many dyes, many probes, and
Yeah.
many antibodies. So we have some solution for that. And then I have one question, Q&A. Okay, so the question is some more information on how we make this high-speed imaging. So so you are correct. Instead of a resonant scanner or spinning disk, we are using a rotating polygonal mirror scanner. So basically, this is the polygonal mirror with very high speed rotations. And then compared to the resonant galvo scanning, it can provide very uniform point, laser scanning inside the field of view, imaging field of view, and then we can achieve the uniform, excitation illuminations over the entire imaging field of view. Okay, so this is a point scanning microscope, but we are using this rotating polygonal mirror to achieve high speed imaging. Yeah, like this.
And then all the post image processing or post processing, those were done using the software.
Yes. Yeah.
That's amazing.
Yeah. So we are providing our own software, but it's based on this work.
I see. So without the processing, the raw video, it looks like the one on the right.
Oh yeah, this is a raw video. So
I mean, even that's not too bad.
yeah, that's true. Yeah.
Oh, that's so fascinating. I know, well, it's 4 o'clock, so I don't want to take up any more of your time.
Yeah, so if you have any more, remaining question or question that comes up in your mind later, then don't hesitate to send us an email. We'll be very happy to answer any question
And
you
the
have.
email address would be information@ivimtech.com.
Yes.
Is it correct? Yes.
Yes, it is. Here. Yeah, the information at ivimtech.com.
Well, Dr. Kim, I sincerely thank you for this amazing webinar, sharing the technology that you guys have developed over the years. And I know it's ungodly hour in Korea. It's very early.
Yeah, no problem.
So I thank you very much. And I am very excited for this technology that as a person who struggled with intravital imaging before,
Okay. Yeah.
I think this would make imaging a lot simpler and post image processing easier as well. So I really hope that I'll be able to host a demo here at Baylor.
Okay. Yeah. Sure. Yeah, and then also, I can share the PDF version of my today's slide, so that may help
Oh,
you to recall.
that'd be fantastic. Yeah, and I can distribute.
Okay.
Oh,
All right.
there are more questions now.
Oh, okay.
Can you provide some information on how you perform the high-speed imaging? most laser scanning image scanning systems use a resonance. Yeah.
And this one I answered. And then
I think yes.
Gary,
Can you
motion
say more about the
correction.
motion correction?
Well, okay. So do you have it would be better if you have a more specific questions, but yeah, the idea is quite simple, very straightforward. Because we are capable of very high-speed real-time imaging, so our solution is we just recorded the short video, like 1 second or 2 seconds, of this, like this, as I show in here. So we do this 1 or 2 second video recording, and then at the same time we analyze the frame by frame, tissue motion, like frame 1, 2, 3, 4, and then we can just see how this individual cell moves frame by frame, and we do the frame by frame image registration to compensate this motion between the frames. And then we average it. And then we accumulate the each image registered frames, and then generate this high SNR images. And then this function is already integrated into our software and hardwares. So this is the real-time, video captures, image screen capture. So we just turn on the motion compensation and press this capture button, and then this is the compensated images. So this is the and then just 2 or 3 seconds, and then we can generate these images.
So this function, you turn it on while you're imaging.
Yes, yeah. This is not post processing. So you can do it on site while you are doing the imagings.
Oh, I see.
But when you do this, basically we are losing the temporal resolution to because we need 1 or 2 seconds of video recording. Okay, so to keep our temporal resolution of 30 to 50 frames per second, then we need to do this post processing. So we have 2 options for the noise reductions. One is the in situ, image registrations, the motion compensation, or this post processing for the noise reductions. So depending on purpose, so we use both.
Yeah, depending on what you're interested in, also matters.
Yeah,
I suppose. And then so for all these images or videos you acquire, I mean, videos are essentially a series of images put together.
Yes.
You can do like intensity, a very simple, very basic analysis. Right?
Oh yeah, of course, yeah.
Very simple. Oh, fascinating. Cool.
Right.
Yeah, if there are no more questions, I would like to thank all of you again, and thank you, Dr. Kim and
Thank you.
Team IVM Technology in South Korea. I really appreciate this. This is like a pre-Christmas gift for me, getting to know this system.
Okay,
So
great.
thank you.
Thank you. Thank you, everyone.
Yeah, I wish all of you happy holidays.